talk about this beautiful thing. Here are the three molecules of life. You guys got this yesterday, I think. Yeah. Is that right? Yeah. Okay. I've made these mistakes before. Here's the difference between glucose, galactose, and fructose. Oh, really?
We got the one that says sugar 1 and sugar 2. It doesn't say.
Oh, let me see it. I'm sorry. Well, let's just do this one then. Let's just take notes on this one. I'll print it for you on Monday. We got a lot to do, so I already have this one set up, so let's just do that. Sorry, messed that up. So, but it's still useful to look at this. Let's take a look at this. Let's do this together. And again, I'll give you the hard copy on Monday, and you can do it. We'll do it in class.
What is the title?
It's the same concept, but it's another different project. Biological molecules, building blocks of life. All right. So let's take a look at carbohydrates first. Here's 3 monosaccharides. Monosaccharides. Why do we call them monosaccharides? And there it is. There's only one. We'll look at polysaccharides in just a minute. Is it time to, what's going on? Oh, it's just. All right, so mono means one, saccharide means sugar. Let's do a comparison. Let's take a look at it. Let's say you group from you. I'm pointing that side of the room, that 1/3 of the room. Raise your hand. All high. From here, in that direction, you guys count how many carbons and how many oxygens and how many. I'm sorry. Let me undo that. How many hydrogens and how many oxygens? That's what I want to know for glucose. From you to you guys, I need you guys to count how many carbons, how many hydrogens, and how many oxygens. Go ahead. Start counting. Sorry, I'm sorry. And then the rest of you guys on this side, count the same thing for these. Go ahead. And write it down. I heard 6, 12, 6. If you remember the rule, 1 to 2 to 1 ratio, and you know there's 6 carbons, everything else falls into place. What about galactose? Same. C6, H12, O6. What about fructose? Same. C6, H12, O6. They're all C6H12O6, but they have different arrangements. Look at this. This is OH. This is the same. This is down. That's the same. This one's down, but then this one's what? Up. That makes a big difference. Not to me and you, but to your, well, it does to me and you, your body, but to the chemistry of these two, the chemistry of these two things is different. Just because this one points up instead of down, just because when it was forming, instead of twisting in a certain direction, to the right, it twisted to the left, that's it, done. It's different. This is almost the same, but instead of connecting oxygen to the end carbon, it connected oxygen to a middle carbon, to this carbon here. This part sticks out. That changes this from glucose or galactose to fructose. This is a 5-ring structure. It's still a hexose, but it's a 5-ring structure. It forms a pentagon. It's a hexose because it has 6 carbons. But they're very different. This does damage to your liver if you eat too much of it. Fructose. Glucose is not a big deal for your digestion, but can obviously lead to diabetes, and diabetic people like myself have a hard time dealing with it. Getting it out of the blood, we digest it fine. galactose can be something that could be hard to digest if you're missing a specific enzyme. So when we're talking about molecules, their shapes make a difference. As I drew yesterday at the end of the day, I drew keys, and I reminded you that if your key just changes slightly, it works in one lock, but not another. So you have to be careful. These small changes make a big difference. So that's a carbohydrate. Those are 3 monomers, and we'll look at polymers in a minute. This is a lipid. You almost want to say it's a polymer. It's actually, this is a triglyceride. Each of these is a fatty acid. A fatty acid. A fatty acid is an acidic group. Why do I call it an acid? I know my functional groups. And because of that, I know what carboxylic acids look like, what the carboxyl group is. The carboxyl group is a C double bond O. Where do you see that anywhere in here? If you're lost, it's because you don't remember what happened 20 minutes ago or half an hour ago when I wrote it on the board. What's a carboxyl group? Look at your notes. You wrote it down. If you didn't write it down, you're not taking good notes. What's a carboxyl group? C double bond oxygen, single bond oxygen. Do you see that anywhere on this page? In this lipid section? Do you see it? Yeah, I see it. Do you want me to highlight it? Has everybody had an opportunity to look at it?
Yes.
Okay. Here it is. C double bond O, single bond O. Do you see it? C double bond O, single bond O. C double bond O, single bond. Remember when you were elementary school, or when you were a little kid, you went to eat at Denny's with your mom and dad, and they said, "Where's Waldo?" And they said, the word, you had that little game, that word circle thing. You used to circle the words with crayons. You ever do that? Well, this is what you're doing here. Where's your C double bond O? Oh, where's Waldo? Well, there it is. This is called a carboxyl group. They form carboxylic acids. So this is what makes this an acid, a fatty acid. Why is it fatty? Because of this, what is this? A long chain of what? Mostly what? Carbon and hydrogen. Look at that chart I gave you yesterday. Mostly carbon and hydrogen. Also known as a hydrocarbon. Long chain, we call this fats. That's what a fat is. So this is a fatty acid because the acid. There's 3 fatty acids, so we call them tri. There's 3 for tri. When you go to a doctor's office, one of the first things they look at is your your triglyceride level. You'll see it on tests. Ask your parents to look at their My Chart. You look at their blood tests, and one of the big blood tests they do is a triglyceride level. This is a triglyceride. Now, why glyceride? Why is this a thing? Well, it turns out that this little tiny, let me use white, I guess. Here, this 3 carbon, usually OH, there's probably an OH here. Probably drawing this wrong, but I'm just going to go ahead. This thing here is called glyceraldehyde. You'll see it pop up throughout the book reading. You'll see it pop up in all of biochemistry. These 3 little carbon, in fact, this is what plants make. They put 2 3s together to make a 6. What have you heard of that's 6? 6 carbons, 2 3s equals 6. What could a a plant put together 2 3s to make what? 2 3s can make a 6. So plants will take these will make glyceraldehyde, and they'll end up making a glucose molecule. All right, so it's called glycerol. Oh, they actually had it here already. I shouldn't have called it glyceraldehyde. It's a glycerol because of the alcohol. Sorry about that. So there it is. It's actually already drawn for you. Glycerol. It's drawn for you here. 3 carbons, 3 OHs. Fatty acids are any of these long chains. You can see we don't have to, obviously nobody wants to draw these out. So what you do is you put them in parentheses, CH2 times how many? 12 of them, with the end being a CH3. That's 1 fatty acid. That's probably this one because it's the smallest one. Here's CH2 times 7 with more CH groups, another 7, so there's a branch here with the end cap being a C. So if we drew it, that's this one here. What is the difference between this one here and this one here? This one and these two. What's the difference? That's right, the double bond versus the single bond. Do you see that? Where's Waldo? I found him. So double bond versus a single bond. If you have a double bond, we call them unsaturated fatty acids. If they are all single bonds, we call them saturated fatty acids. When the fatty acid is unsaturated, what that means is, let's look at what they look like when they're saturated. That means that all the bonds have a hydrogen. That's what saturated means. They're full. All the carbons have a hydrogen on them. All the bonds have a hydrogen or a carbon or an oxygen. There's no bonds. They're just kind of forming 2 bonds. If you have an unsaturated, what happens is you have C, you have a double bond somewhere, so that you have a carbon You have a carbon that does not have a hydrogen attached. So they're unsaturated. Well, what's the big deal? Well, there's a big consequence to that. These saturated fatty acids, they form long chains that can layer one on top of the other. Now, they don't form hydrogen bonds. They don't. But because they're heavy and they're long, they're very long. As you can see, they're 20, 30 atoms long. They don't easily turn into gases. It's not like carbon dioxide or oxygen that are small and nonpolar. These are very big and nonpolar, so they don't turn into gas easily. And because of that, they can lay down, water can be surrounding them or not, they can lay down in flat sheets and they form solids. Where the other ones, the unsaturated fatty acids, they can start to go long, but then this makes them kink, makes them bend. And because of that, they find it harder to lay flat on each other. They'll lay down, but they won't lay flat. So these guys here end up being mostly, they tend, I'm going to use the word tend, to be liquid at room temperature. And these down here tend to be solids at room temperature. Let's just do temp. So these tend to be butter or lard, right? Those are things that tend to be solid at room temperature because they have a lot of saturated fatty acids. Where these up here might be corn oil or olive oil because they tend to be liquid at room temperature. That's why olive oil and butter have two different states of matter at the same temperature. That's why when you're done cooking with corn oil or olive oil, the pan cools down. What's in the pan? When you're done cooking, frying something in corn oil, what's in the pan after it cools down? It's a liquid. What's in a pan if you cook with butter after it cools down? Or you fry a pork chop in a pan and you take your pork chop, you go eat it, the pan's time to wash the dishes. What do you find in the pan? What is it? Solid. So the difference is one's saturated, the other's unsaturated. Now, health-wise, how does that make a difference? It turns out, I'm really simplifying this, but it turns out that these can lay on each other very easily in your blood too, and they can start to lay and form these things called plaques. They get chemically changed. So if your blood vessel looks like this in that bloodstream, you over time, as you're eating saturated and other types of fatty acids, the theory is that these things turn into these little plaques. They start to do what? Start to block your arteries and veins. And they can block tremendous amounts before they become a problem. And then you get a small blood clot because somebody punched you in the nose, or you bumped your knee or something, and that blood clot comes along and it gets stuck in that artery or vein. So that means you don't get any blood flow through that vein or artery. We call that a thrombosis, right? It could be a heart attack. It could be a stroke. It could be a lot of things. So as you get older, if you've been eating a lot of these, it has to be a lot. It turns out saturated fatty acids is good in small amounts. We need it, but it's the unsaturated fatty acids that actually tend out to be the good part, the good kind of fatty acids you need to be eating. So you reduce the tendency of your body to form these plaques. So I'm hoping I'm making a connection for you why this is important. This idea of saturated versus unsaturated fatty acids. Now, proteins are made of amino groups, and this is a drawing of an amino group. The amino group is here. I'm going to identify, here's Waldo. There's an amino group. There's an amino group. There's an amino group. And it says amine group. I'm sorry, amine group. What's on the other side? What's on the other side? Carboxylic acid group or carboxyl group. So I got amine on one side. I got an acid on the other side. What am I going to call this? Amine on one side, an acid on the other side, amino acid. You need to know what these look like. Sticking off the side is a variable. It's not an element. It's a variable. We call it the R group. It sticks off the side. There's 20 different possibilities. There's 20 different R groups here. Okay? If it has a carbon with a sulfur on it, we call it cysteine. That's the amino acid cysteine. If it's just a carbon and 3 hydrogens, here for the R group, we call it alanine. This is nonpolar. This is small. This can form sulfur. If you have a lot of cysteine in this protein that you call hair, then your hair is kinky. The more cysteine you have, the more genes you have that say put more cysteine in this protein, right? It's a gene. Then the curlier your hair, the kinkier your hair. If you have less cysteine in that protein, then your hair is straighter. That's it. That's the only difference between really curly hair and straight hair is how much cysteine you have in it. Because the sulfur makes these types of bonds that are like hydrogen bonds. We call them disulfide bridges, but in any case, that's the point there. So which amino acid? There's 20 different amino acids. There's amino acids that have ring structures. There are amino acids that are acids. There are amino acids that are bases, basic. There's amino acids that have formed these aromatic rings. They're really large amino acids, really small amino. The smallest amino acid is the same structure, the amine group, the carboxyl group, the H group. And here you have an H. It's alanine. It's the smallest amino acid. So there's 20 different ones. That allows proteins to be very complicated. It would help you understand what's going on if you were paying attention to this instead of being on your phone. That would be very useful. Now just focus on the job in front of you. All right. Here is the sugar ribose or deoxyribose. Which is this? Ribose or there's two possibilities. It could either be ribose or deoxyribose. It's not easy to tell. You probably won't remember. I don't think I mentioned it recently. You're looking at this carbon right here. You're looking for what? What are you looking for? Whether or oxygen or no oxygen. If it's no oxygen, it's what? Deoxy. And if there is an oxygen, then it's ribose. All the oxygens are usually written. The hydrogens, most of them are never written in these drawings. Okay? Which of these is it? Deoxy. This is deoxyribose. This is DNA. All right? So we're looking at DNA. DNA nucleotide is made. These are monomers, by the way. These are monomers. You can put these together to make disaccharides or polysaccharides. These are the monomers for proteins. You can make long polymers called proteins or short polymers called proteins. There are dipeptides or tripeptides and polypeptides. And nucleic acids obviously form you put a bunch of these together, you get billions long, called DNA, right? A DNA nucleotide. This is called the nucleotide. And there it is, it says nucleotide, right?
How would you describe the position of the oxygen in a ribose?
Good question. So with all ring structures in chemistry, you start with the zero. With at least organic chemistry. You start with the zero, all right? And you go around clockwise. This is position 1 prime. This is position 2 prime. This is position 3 prime. This is position 4 prime. And this is position 5 prime.
So you would say on the 2 prime?
2 prime is correct. And that's how you number these. There's only 5 carbons, by the way. Both ribose and deoxyribose are what we call pentoses. There are only 5 carbons. All right? On the 5 prime, this is really important because it turns out that because there's 2 different things happening, because and I'm going to draw it out here as a block in a minute. Over here you have the phosphate group, that's part 1 of a nucleotide. You have the ribose, the sugar part, that's part 2 of a nucleotide. And then you have the nitrogenous base, that's part 3. Do you see why it's called a nitrogenous base? They call it nitrogen base. Why do they think they call it a nitrogenous base? It's really simple. The questions I'm asking you are generally simple. Why do they call it nitrogenous base or nitrogen base? Because it has nitrogen in it, and it's a base. That's all. Honestly, scientists, although we try to be imaginative, are generally try to be really simple. Yeah.
Would you say that it's on the part two?
Someone close that door, please. What is this?
Describing the position of the ribosome. Are they all, is that,
This would be, what I would say is the nitrogenous base is on the first 1' of the ribose. Always. The base is always attached to the 1'.
I thought you said it was the sugar, the middle one was the sugar.
This is the sugar, yeah.
So it's always on the sugar?
It's always connected to sugar, yeah.
Okay.
The base is always connected to the 1' of the sugar. On the 2', I can look for the oxygen to determine if it's a ribose or deoxyribose, but it's really not involved in any kind of chemical reaction. It's just having the OH or the H forces, because of geometry, forces the nitrogenous base to be either T or U. That's the only issue with this second carbon. Then this OH is always on the 3rd. This is incredibly key. And the phosphate's always on the 5'. So when we get into DNA replication and DNA structure, this becomes very important. Let me show you why. So you have this phosphate group. It's attached to a carbon, right? That's attached to, as someone just asked, to a ribose or deoxyribose, in this case deoxy because it's sugar, which is then attached to a hydroxide group, which can then be used to attach to what? Can anybody make a guess? What would you attach then to? What would you attach to this? Actually, this leaves. What would you attach to this? I'm just going to go ahead because we're running out of time. Another phosphate, which is attached to what? Another carbon, another ribose or deoxyribose, which is attached to what? A phosphate, which is attached to what? Another deoxyribose, which is attached to a phosphate. It's attached to another deoxyribose, and then attached to another phosphate. Yeah, I was gonna keep saying it till a lot of people started complaining. That's all. Yeah, it's a chain. It forms a really long chain. I mean, really long. When you leave the room, you can look outside and that beautiful poster that I bought so long ago. And it's just this long chain. It's just this long chain. All right? But because there are two sides, because there's a 5' and a 3', because they always attach this way, because you're always attaching the phosphate to the 3' of the previous nucleotide, because that's the case, you have a 5' side to the molecule, all right? And you have what's on the other end? What's on this end? A 3'. These molecules have direction. They go from 5' to 3'. That's how you attach them. What does that allow us to do? My God, guys, this is amazing. This is so stunningly crazy that because there's direction, you have order, you can write a book. This becomes a language. Because there's differences in between them in the nitrogenous base, you can have an A, the nitrogenous base T, the nitrogenous base G, the nitrogenous base C. It could be anything depending on, and it can be billions of nucleotides long. Now you have a book, which then can be defined into genes, and then genes can make someone tall or short or have wings or have be a jellyfish or a fish or an octopus or a human or whatever, because there's a series of letters that has direction. If I had a bunch of letters and there were no direction, I didn't know which was left, right, which way to read it, then there's no way to have a language. You can't have a language without direction. You can use anything to form a language. We're used to using letters, but in Arabic, the letters are more like script, right? Like drawings. In Chinese, they're symbols that mean ideas. They don't even mean specific words. You put the different symbols together to form different words. Like, I don't know. I don't know Chinese, but I know that much about it. So the same in Japanese. In Korean, they're more like letters. In the ancient Mayan calendar, when the conquistadores, my ancestors first came, some of my ancestors first came to the New World, and they killed the Native Americans and took over the Aztec temples and we killed everybody. We killed them with this. We gave them smallpox blankets. We shot them. We wanted the gold. That was it. Sorry, you're dead. We want it. I'm not saying it was a good thing. I'm just saying it did happen. So bottom line, when some of my ancestors came here, they brought their priests, and the priests tried to figure out what the heck was going on in the temples, and they went to the libraries. They found a bunch of strings with knots in them hanging on sticks. What do you think that was? It was their language. They were their books. Their books were written in strings, top, down, outer, in. Direction, order, gives you words or math. They kept all their math there. An abacus is not numbers. They don't use numbers. They use little beads on a string, and they move them around. And they, in their minds, it means the same thing as 1003 for you. So language can come in chemistry as well.
Can you zoom in on the paper at the bottom?
Oh, I left this like that. Zoom in where? The paper at the bottom.
Yeah.
All right, good. I know it was a lot, but I think we had a good class.
Can you do a little bit
Huh?
A little bit more?
Yeah. I know it was a lot. Thank you for your patience. I'll try to minimize these days, but it is AP Biology, guys. There's a lot to cover. And it's either you read it on your own or I explain it. And me explaining it means language. Sorry. So here it says, use Model 1 to show which atoms are present in each type of molecule by listing the symbol for each atom included. Carbohydrate has been done for you, CHO. So if we think about it, carbohydrates are CHO, but remember let me go ahead and just kind of put it off to the side here. The carbohydrates are one carbon to two hydrogens to one oxygen. I don't like that it looks like a 10, but so let me make it one oxygen, right? So one carbon to one hydrogen to one oxygen. So that means, again, for every one carbon, you're going to have two hydrogens and one oxygen, all right? So remember that, 1 to 2 to 1 ratio. In lipids, you do have some, you're mostly, and let's just go ahead and say mostly carbon and hydrogen. You do have, and I'm going to put in parentheses here, you do have sometimes you have phosphorus. First of all, you do have some oxygen. There's some oxygen, Sometimes there's some phosphorus and even some nitrogen sometimes, but these, the nitrogen is very rare compared to the others. There's obviously a lot of oxygen, and phosphorus is in certain kinds of lipids. But for the most part, when you're talking about lipids, you're talking about carbon and hydrogen, long chains of carbon and hydrogens, or a ring of carbon and hydrogens, mostly carbon and hydrogen lipids. Amino acids, you're dealing with carbon and hydrogen and nitrogen, and some of the R groups, and of course you have oxygen and and the amino acids. Some of the R groups have sulfur in them, but no phosphorus. This is one of those key points. Though lipids do have phosphorus, not all lipids do. There is no phosphorus in amino acids, and there's plenty of nitrogen in amino acids. Plenty of nitrogen. Nucleic acids also have a lot of nitrogen. So you have, with nucleic acids, you have carbon, you have hydrogen, you have oxygen. That should be fairly reasonably understood by now when you think about the fact that nucleic acids do have that sugar, either ribose or deoxyribose. They have the nitrogenous base, and then they have that phosphate. All right. So remember, that's the nucleotide. That's the monomer sketched out there. So when you're dealing with nucleic acids, you're going to have carbon and hydrogen and oxygen, because you have a carbohydrate as part of the structure of the monomer, but you also have nitrogen and phosphorus. And let me rearrange that and change that color and say nitrogen and phosphorus. So when you see nitrogen, you're going to find nitrogen in places like amino acids, nucleic acids, and even lipids sometimes. But you are definitely going to find phosphorus mainly in nucleic acids. Obviously, lipids, phospholipids have phosphorus in them, but you're really going to find a lot of phosphorus in nucleic acids because it's part of the structure of nucleic acids of DNA and RNA. Amino acids are mainly the main identifier is nitrogen, but then it's going to be hard to tell the difference if you're just looking for nitrogen between amino acids and nucleic acids. It's the sulfur that's going to be the key. If you find sulfur somewhere, then you're probably looking at amino acids or proteins. Now, I'm not going to go back and go over all the components. You have all the notes for all the different monomers, but you should know for the exam, like for the biomolecules, of lipids, nucleic acids, and proteins and carbohydrates. You should know the monomer and polymer of each. So in lipids, you do have fatty acids that can form. Fatty acids do form parts of a triglyceride, but honestly, you have to say there are no monomers or polymers, because when you're talking about a polymer and a monomer, you're talking about taking like a Lego, like I keep using the Lego blocks as an example, taking a bunch of Lego blocks and putting them in locking them together. In the case of molecules, it would be covalent bonds. But taking these Lego blocks and linking them together to form something bigger, lipids don't do that. They're not you can't make these giant long molecules like that. Lipids are just carbon and hydrogen. They don't really have monomers or polymers. Nucleic acids, I hope by now you know it's a nucleotide. At this point, the test is on Monday. I'd expect you guys know that a nucleotide is a monomer, and the polymers are either DNA or RNA. And the proteins, you have amino acids, are the monomer, and the polymer are polypeptides. And there's whole long days of lectures and information. I told you to make index cards on all of these. This is stuff that you need to know. Carbohydrates, there you're talking about monosaccharides. Let me zoom in and write it. So you're talking about monosaccharides. And the polymers, polysaccharides. The monosaccharides are, remember, glucose, galactose, fructose. Those are the ones we talked about in class. And, of course, ribose. And, of course, deoxyribose. Remembering that deoxyribose and ribose are, let me go ahead and separate them. Deoxyribose and ribose are pentoses. There are 5 carbon sugars. And fructoses, galactoses, and glucoses are 6 carbon sugars. They're monomers. Now, polysaccharides, you also have some disaccharides that we've discussed. And some of the disaccharides are table sugar or something called sucrose. Do you need to know that sucrose is glucose and fructose? If you know that, that's great. You do not have to know it for the test. I'm not going to ask you such a question. And, lactose is another disaccharide we talked about. Do you need to know that it's glucose and galactose connected together? No, not really, but it's nice that you know it. It'll help you in the long run, but in the years to come. But right now, for this test, no, you do not have to know that. You should know that sucrose and lactose are disaccharides. It means two sugars linked together. 2 Lego blocks. The prefix di means 2. All right, so 2. All right, whether it's glucose and fructose, or whether it's glucose and fructose, or glucose and galactose. If it's glucose and fructose, it makes sucrose, table sugar. If it's glucose and galactose, it's lactose, it's milk sugar, etc. And then the polysaccharides we discussed are cellulose, which is, you call it wood, right? paper, pencils, wooden structures. The sugar made for structure in plants, not digestible by anything except for bacteria and fungi with special enzymes. It's the only way to break down cellulose. It's so carefully put together. Another polysaccharide is starch, and that's found in plants. We eat it. And finally, another polysaccharide is going to be glycogen, and that's in animals, in the liver of humans and mammals, but in animals. All right, so 3 polysaccharides we discussed: cellulose, starch, and glycogen. And then you have 2 disaccharides we discussed: is sucrose and lactose. You should know that cellulose is in plants, that it's for structure. You should know starch is in plants, it's for energy storage, and glycogen is in animals, or energy storage. That kind of thing we should know, and that's kind of a quick, and yet one more quick review. But if you wanted to tell the difference between these 4, these are the elements that are found in each one. Which type of molecule includes the example of long-chain carbon backbone? Long-chain carbon backbone. Now it says carbon. If it said carbon and phosphate, then it would be DNA. But it says long chains of carbon as the structure upon which everything is built, the backbone. You'd have to say lipids, right? Why? Because it's carbon. It's mainly carbon and hydrogen. It's mainly carbon and hydrogen. Yes, there are other elements involved, oxygen, phosphorus, and nitrogen sometimes, but carbon and hydrogen being the main component. All right. In a molecule referred to in the previous question, which is the dominant element attached to the carbon backbone? And that it would have to be hydrogen, right? As I said, it says dominant element, so it's carbon and hydrogen, right? The carbon backbone with hydrogen attached. Fatty acid chains and lipids is often referred to as a hydrocarbon chain. Hydrocarbon, hydrogen and carbon. Discuss with your group why the chain is given the name. Write 1-sentence definition of hydrocarbon. Long chain of carbon and hydrogen. Now, I have already given you all this information in class, but again, I just want to make sure that it's all written down in order and there's no confusion about what the right answer is. Which molecule has a central carbon with 4 different components around them? Central carbon with 4 different components. Well, I drew it in class. Again, giving you all the answers. You have a central carbon. Then you have attached to that central carbon an R group. On the other side, there's a hydrogen. Then on one side, you have a carboxylic acid group. And on the other, let me use a different color. On the other, you have an amine group. So you have central carbon with 1, 2, 3, 4 different groups surrounding it. We call that an amino acid. Amino acid. Right. the amine group and the carboxylic acid group, helping define that word. Which molecule has sugar and nitrogenous-based phosphate group? Now here again, I'm going to draw it out over here because there's more room. Well, I already drew it. I'm not going to draw it out again. Well, I guess I might as well. Has a sugar. Yes, there's hydrogens and carbons. It's a 5-carbon sugar again. 1, 2, 3, 4 prime, 5 prime. To that 5th prime carbon, there is attached a phosphate group. And on the other side, a nitrogenous base. Right? So as a nitrogenous base and a phosphate group attached to a sugar, what the sugar can be ribose in DNA, and can be deoxyribose in RNA. In DNA. So DNA is deoxyribose, and I don't even know what I said a minute ago, but RNA is ribose. Okay? So the answer is nucleic acids. The two main nucleic acids that we talked about, the only two we talked about are ribose and deoxyribose nucleic acids. Discuss with your group members some similarities among all 4 types of molecules. List as many as you can. Well, they all have carbon, hydrogen, and oxygen, with carbon being the center component of just about every one of them. You make them all the same way. Let me be neater because I know I'm going to hear it if I'm not neat. Make them all with dehydration synthesis That's 2 similarities. They all use the same group of elements to be formed. All of them use the same group of elements, which is schnapps, with different percentages for each one, right? Carbon, nitrogen, oxygen, phosphorus, and carbon, nitrogen, oxygen, phosphorus, and sulfur. All right. What is the chemical formula of the first carbohydrate molecule shown? Well, if you look at the first one, it's C6H12O6. 3 structural groups shown. Do all amino acids have, what 3 structural groups shown do all amino acids have in common? Well, they all have the carboxyl or carboxylic acid, the amine, right? And hydrogen. They all have all 3 of those. / central carbon. The difference is, of course, the R groups. There are 20 naturally occurring amino acids, and each one only varies in the structure of the R chain. There's that R group or R chain, right? 2 amino acids shown in model 1. What are the R side chains in each? Well, one is CH3. That's the R chain. That means that's, to draw it out, it would be 1 carbon. Let me draw it neater. Trying to be neat and move it quickly at the same time is very difficult. CH3. So you have a carbon with 3 hydrogens attached to it, sharing. Remember, carbon always makes 4 bonds. And this last one goes to the center carbon, right? So that's CH3. That's how we would note it. The other one is CH2SH. So that's one carbon. Now remember, these are R groups. So to be clear, when you look at an R group, you're looking at this group sticking off. So CH3 would be sticking off here. So you have amine, center, carbon, hydrogen, carboxyl group, and then you have CH3 sticking out here. All right. So that being said, what does that look like? Well, that's going to look like, well, you have a carbon. You have to that sulfur. Here you have a hydrogen sticking out, and the sulfur has a hydrogen sticking out. So that's all. So you see the only difference between these two is that you have a sulfur here instead of a hydrogen, 3 hydrogens. Carbon still has 4 bonds. During chemical reactions, the bonds of molecules continue to be broken and reformed. To break a bond, energy must be absorbed. When bonds are formed, energy is released. If more energy is released than absorbed during a chemical change, the process can be used as a source of energy. The general rule for processes such as respiration, carbon is the more carbon atoms that are in a molecule, the more energy the molecule can provide to the organism when it is used as food. So we did an experiment in the lab. We did a lab where we took water and we broke it. So we took oxygen and 2 hydrogens, and we snapped these. We snapped these bonds, and we used electrical energy to do that. In the form of a battery, right? We inputted electrical energy, broke the bonds. Then we just added just a tiny bit of energy, And what we did was we formed water. We put it back together. When we form water, we put energy in to break water up. When we form water, energy came out. And it didn't sound like a lot because it was just a few million molecules. When you use more material, you get more energy. But I think that's obvious. Like the difference between a firecracker and a stick of dynamite is how much explosive material you have in it, right? The more you have, the more intense the explosion. So our explosion was tiny because your parents want you back in one piece, and I want you going home safe. So we made those tiny little explosions, and that released a lot of energy. When water was put back together, I don't know why I drew it that way. Let me erase that. So when we put water back together again, right? We took that. We used electrical energy, and what we formed was hydrogen gas, and then oxygen gas. We collected it in a test tube, right? And we know that there's a 2-to-1 ratio. So this is 2 hydrogen gases for every one. So we had a lot more in the hydrogen tube than we did in the oxygen tube. And so when we made that hydrogen gas, and then we added some oxygen from the air, oxygen gas in the air, and let me just put in parentheses down here, from the air, right? And we allowed the hydrogen and that oxygen to mix, and we added just the tiniest of little sparks, a little bit of heat from our flame. We got an explosion, and then what formed was water. And it was liquid at room temperature. So what we did, what is it that one of the things you should have gotten from that, or hopefully you got from that, is that we needed here at this stage, when we were going in one direction, we had to input energy. And here, when we reversed it, we got energy out. Now, if we got more energy out than we put in, then we're making energy. We can use that energy to run reactions like our thoughts, our heart beating, etc. And that goes for any chemical reaction. You're either putting energy, you get a net energy in, or you're putting net energy out. So when you have a long chain of carbon, like in fatty acids, for instance, in fats, and if they're the longer the chain, the more bonds you have to be able to break apart, to use. The more energy you have, the more of these bonds that you can break and then reform in the form of carbon dioxide. And when you do that, you get energy out, and boom, you can use that energy to run the different things that you need to do in your body. So fats are one way we can do it, but we can use water as a simple way of looking in a very simple ratio, and I think fairly dramatic way. We can see energy coming out, energy putting in, electrical energy going in, and we get out that explosive energy when we make water. Here it says, using the information above, is this a carbohydrate or lipid? More likely a good source of energy for an organism. It's going to be the lipid, right? Because the lipid has a lot more bonds that you can break. And so lipids are much better at holding energy. There's a lot more energy storage with lipids than you can with carbohydrates. So here's a second model. Here again, glucose and fructose. Here's amino acid 1, amino acid 2. In both cases, we're putting them together. All right, I've drawn this out for you numerous times, but let's go ahead and see exactly what's going on in a professional drawing here. You have OH and OH, and they're coming together and they're forming a bridge. This oxygen is going to be used to bond to this carbon, remembering that at each stage, at each one of these places where there is no C, there is a carbon. So this is carbon 1. It's actually, it's complicated, but this is actually carbon 1 prime, 2 prime, 3 prime, 4 prime, 5 prime, and 6 prime. And this is carbon 1 prime, 2 prime, 3 prime, 4 prime, 5 prime, and 6 prime, right? Now, why were they numbered that way? They were numbered that way before they closed in a ring, and after they closed in a ring, when the oxygen forms that ring with the second carbon here, it forms fructose. If it forms the ring with the first carbon here, then it forms glucose. Irrelevant, that's not the point. The point is we're going to use this carbon, we're going to connect this ring to this ring, all the carbons, I'm going to put a purple dot at each carbon that's not labeled with a C, remembering that every time there's a line that comes together, they form that is a carbon. And if you count them, each carbon has 4 bonds. 1, 2, 3, 4. 1, 2, 3, 4. You can go around and count them. Even this, it says CH2OH. If you broke it up, if you opened it up, this is kind of a condensed look at it. If you looked at this, right, use a different color. And let me use green. So if you looked at this, it would look like carbon, then an OH group, then two H's. And then this whole thing would be attached to this carbon. So that's what CH2OH means there. So what you see is if you count them, there's a single bond, there's 4 bonds to this carbon, there's 1, 2, 3, 4 bonds to this carbon, etc. Okay then. When we look at the combination, when these two coming together, what you get is an OH and an H. An OH and an H coming together because something has to leave. Remembering oxygen can only make 2 bonds. Let me before I do that, there's a bond between the oxygen and hydrogen, a bond between the oxygen and hydrogen, and then there's a bond between this oxygen and this carbon, this oxygen, this carbon. That forms 2 bonds for oxygen, 4 bonds for carbon, 1 bond for hydrogen, right? So when we draw the line, something has to leave in order to get these 2 molecules, these 2 monomers, these are called monomers or monosaccharides, to come together. We have to have something leave, and what generally leaves in these reactions is water. So water comes out, and what gets put together is that oxygen still needs to make 2 bonds, but instead of this oxygen being bonded to this hydrogen, this oxygen now binds to this carbon. And so oxygen has 2 bonds, and each carbon has 4 bonds, 1, 2, 3, 4. And now these 2 are connected. And we name this sucrose, right? Table sugar. And of course, we got water out. Where'd the water come from? The hydrogen and the hydroxide coming together making water. Exactly what we did in the lab. All right, so when we're looking at an amino acid, these 2 are coming together. Again, if you're going to put this amino acid and nitrogen can make 3 bonds, 1, 2, 3, that's full. Carbon can make 4 bonds, 1, 2, 3, 4, that's full. How do you get these 2 connected? Well, you got to get rid of something, like we've said numerous times. This hydrogen, these hydroxides get together. They form water, right? They leave in the form of water. So as they leave, in the form of water, what you see connected between the 2 is the NH group of 1 gets connected to the carboxyl group of the other, and you get carboxylic acid on one side and amine on the other, and there's a connection between the 2. We call that a dipeptide. This is sucrose. Both of them are 2 monomers connected together, remembering that a monomer of a protein is amino acids, so 2 amino acids coming together would form not a polypeptide, poly means a lot, but a dipeptide meaning 2. Sucrose is a again, the 2 again, disaccharide. Di meaning 2, saccharide meaning sugar. Now, when you're talking about lipids, again, carbon-hydrogen backbone, mostly carbon hydrogens. I think if you look at this, and no one could argue this, mostly carbon and hydrogen. Even the glycerol molecule is mostly carbon and hydrogen. Has more oxygen than a fat does. There's a double bond oxygen here. This carbon is a carbon double bond oxygen, single bond oxygen. So it used to be a carboxylic acid group. The H left, right, and then the OH from here left, and the H from this hydroxide left. Together they form this bond. I've drawn it all out in your notes. I'm not going to do it again. All this I've done in your notes before, but I can't just redo everything, so I'm going to go on. So if you have this long chain of carbon, that's one. Now you got two other spaces. You can take this fatty acid. I'm going to go ahead and draw this out, even though I've done it in class. I just want to point out that this means double bond oxygen, single bond OH, right? Single bond carbon with two hydrogens repeated 14 times. So it would be over and over again 14 times. That's all that means. It's just a shorthand to let you know that this is repeated 14 times. So instead of doing that, I'm just going to put dot, dot, dot. Then you have the final carbon with 3 hydrogens because every carbon needs 4 bonds. So that's your fatty acid. And you're going to take that and you're going to connect it to this. How are you going to do that? How does that happen? It happens the same way as everything else. OH and H leave. Let me zoom in on that. OH and H leave. And what you make is water, right? So you put them together and you make water, so you get this, and that's a diglyceride. This is a monoglyceride, meaning one glyceride molecule. It's a diglyceride, and then you can have a triglyceride, but you're never going to have a polyglyceride, for instance, so there's no polymer. That's why I don't even want to talk about a monomer, because the max you're going to make is these 3 together. You're not going to make many like you can with DNA or amino acids or sugars or carbohydrates. So, okay, so we're talking about dehydration synthesis. What are the reactants in A? The reactants in A are monosaccharides. Or carbohydrates, if you'd like. What are the products in A? They're disaccharides. And water. Each of the reactants in A is a single sugar molecule, also called a monosaccharide. What is the prefix? Well, maybe they wanted specifics, so you could say glucose and fructose, and then here you could say sucrose, which are disaccharides, and glucose and fructose, which are monosaccharides, right? Each of the reactants in A is a single molecule, so known as monosaccharides. What prefix before saccharide would you use to describe sucrose? Well, we already said it, and they told you already. It's di, which means 2. What are the reactants in reaction B? They're amino acids. And what 2 molecules in reaction B are joined together? When 2 molecules in reaction B are joined together, or amino acids are joined together, what other 2 molecules are produced? There would be a dipeptide and water. I just want to remind you that when we're talking about this, you're talking about this word peptide, what it means. It means 2 amino acids bonded together. This here, this bond between this carbon and this hydrogen is known as a peptide bond. And because it's a peptide bond and you have 2 amino acids, we call them dipeptides. That's it. So it's a dipeptide that you put together in reaction B. You can go look at the paper on that. And what product do all three reactions in Model 2 have in common? Water, H2O, as a product. Water is always produced in dehydration synthesis. When two sugars are joined together, a new bond that forms is a glycosidic bond. There's two bonds between sugars are glycosidic bonds. The amino acids join together to form a peptide bond. When fatty acids are joined to a glycerol, that bond is called, holds them together, it's called an ester bond. So they actually have names for each of the bonds. As I mentioned, peptide bond. On the diagram Model 2, circle and label the glycosidic, peptide, and ester bonds. So it says on the diagram Model 2, circle and label the glycosidic, peptide, and ester bonds. The glycosidic bond, I'm going to circle it like they ask, and I'll do it in blue. This is the glycosidic bond because it's between two sugars, between the two amino acids. I already did it up in the drawing previously, but I'll go ahead and circle it here. It's the peptide bond. And then the ester bond is going to be here between the fatty acid and the glycerol molecule here. Those are the ester bonds. These reactions can also be reversed, breaking a large molecule into its individual molecules with substance would need to be added in order to reverse it. Well, I'm pretty sure I've told you numerous times, but I'll say it again. Water goes in and breaks them apart. Lysis means to split or separate. What prefix would you add to lysis? If you're trying to separate all these biomolecules, what would be the prefix you would use? If lysis means break, what would you put in ahead of it if you need water to break them? Give you a minute. So word is hydrolysis, using water to break it. Using your answers from the previous two questions, what word is used to describe the reaction that uses water to break up a large molecule? Well, hydrolysis. Oh, I guess I skipped to the point, didn't I? Apologize. Hydro is there. That's the prefix, and together you would make hydrolysis. All right. To break water, to break up large molecules using water. So to be clear, you have a large molecule, you have some kind of reaction, it's reversible, right? It goes in both directions. You can use, you know what, let me draw two arrows here, just because I think it would work better if I did that. So let me draw two arrows. And you're going to use some big molecule, and you're going to break it up into its units. Right? You're going to break up something into a biomolecule into parts. That's reversible, so you can go in this direction and break it apart. Or you can move in this direction and put them together. So when we break it apart, take a big thing and break it apart into little things, we're going to call it hydrolysis, because we're adding water, goes in, MS is used, all right? And then to build it, it's another word for building is synthesis. And we're going to make water. Water's coming out, so we're going to call it dehydration.
I hate that it takes this long. Sorry, I have to start all. I've written it 3 times. Dehydration. Dehydration synthesis uses water. I'm sorry, produces water. It kicks water out. That's why it's called dehydration. So to build things up, use dehydration synthesis. To break things down, you'd use hydrolysis or hydrolysis. You're using water to break things up. You're making water when you build them up. Build them to make something, a large biomolecule. This is a biomolecule. So you're building a big biomolecule, and these are the monomers. Now, let's not even call them that. Smaller units. Why don't I just call them monomers? Because lipids don't really have monomers and polymers. These ester bonds are broken. You do have fatty acids, so I'm not going to get into the discussion about the difference between monomers and polymers. Just know that this isn't, lipids are not true polymers, so let's not even call them that. They are molecules, but not true polymers. All right, let's move on. So it says metabolism is the collective term used to describe all the chemical reactions taking place inside living organisms. So why is water so important in metabolic? I don't know what's wrong with me. Metabolic reactions. What is water? Why is water so important in metabolic reactions? I'm sorry, it's been early in the morning and I haven't had a lot of sleep, so I apologize. So, metabolic reactions. Why is water important? Because it's produced when building reactions or molecules. And it's used in the breakdown of biomolecules. We store excess food in our body, either in the form of carbohydrates, in muscles, and liver, and fat, and adipose tissue, right? So carbohydrates are stored in the liver and muscles in the form of, and you should know because I summarized it at the beginning of this, but I'm going to go ahead and and say glycogen, right? That's how it's stored. And in adipose tissue, it's stored as fat around your belly and different places in your body. You store excess food, excess energy in the form of fat. When your body needs additional energy, it uses carbohydrates source first. Is it quick? So it uses glycogen, which, by the way, is how our feedback loop that we talked about with glucagon and insulin works. It releases glycogen from the liver. All right. Why do you think carbohydrates are used as a source of quick energy rather than fat? Use complete sentences and your scientific terminology in your response. Well, when you consider what we've talked about for the last, I don't know how long I've been on this, but also what we've been talking about for a while now, and what you've read here repeatedly, I hope it makes sense that carbohydrates are, especially monosaccharides, are small let me rephrase that. Monosaccharides are small and can be broken down quickly. Energy can be released very fast. Lipids are bigger, or fats are larger molecules, or longer chains. And take longer to break down. So lipids are great at storing energy, but they take longer to break it down. If you need energy now, you probably want to use a monosaccharide. Luckily, sugars form polymers, which means you have thousands, if not hundreds of thousands, if not whatever, I don't even know the number, of units of carbohydrates, monosaccharides, linked together in a chain in your liver, and plants have them in their and that where you attach them is important, but I'm not trying to specifically draw these in any particular way. I just am trying to emphasize that they're long chains, all right? So, and I mean extremely, extremely long chains, right? So what's cool about that is when you want to break it down, you start to break down the units, and as soon as you split this up, this can be burned quickly, this one can be burned quickly, this one can be burned quickly, and you can throw them into the bloodstream, and they can travel all around the body, and each individual unit can be broken down very quickly. Lipids are much more difficult to manage because they're long chains. They have to be put in little globules called HDL or LDL, and they have to be transported through the bloodstream to different parts of the body. Then they have to be broken. The long chains have to be chopped down. So the example, I mean, something you might think about. It's not quite a great example, but if you think of like a matchstick, how easy to burn is it to break down and use a match to a bunch of kindling to make a fire, versus chopping down a big tree, then chopping the tree down into bits, and then using those bits to start your fire. So that's kind of the difference between monosaccharides and lipids, and at this aspect of the biomolecules, is that monosaccharides are small units and they can be easily broken down, where lipids are longer chains, or fats are longer chains. Look at the two types of fatty acids below, saturated and unsaturated. What is the difference between the two? Remember in our discussion with saturated and unsaturated, if you look at the carbon and they're all the single bonds, all of them are full, either they're bonded to another carbon or hydrogen, we call that saturated. As soon as you have a missing hydrogen, the carbon is an unsaturated carbon. It's an unsaturated fat. That means the carbon does not have single bonds, and not bonded. Not all the carbons are bonded to either hydrogen or oxygen. There's a double bond to another carbon, so it's unsaturated. So the difference, as we've discussed in class, is this. Saturated fats are solid fats like animal fats, lard, butter, whereas unsaturated fats are more fluid and form oils such as vegetable oil. Trans fats are plant oils that are artificially solidified to make them suitable for baking purposes, right? So we can use these, we can change these artificially, and we can make these solid at room temperature, even though they're plant oils. In recent years, trans fats have been associated with negative health issues that are not widely used. Explain the simple molecule term that would have to be done to plant oil to transform it into a trans fat. And it sounds like it's something would have to be nutty to be able to do. But the point would be, you would take this unsaturated and make it saturated. Well, how would you do that? Well, you would do that by adding a hydrogen. If you add a hydrogen there, now you can layer the fats on top. It'll be solid. But without it, this unsaturated fat is kinked and will be liquid at room temperature. But if you add a hydrogen, it straightens out, and now you can layer them out. They become solids, like margarine. Margarine used to be something people ate. I don't even know if you can buy it anymore. They would have a lot of trans fat in it. People thought trans fat was healthier than saturated fat. Well, it turned out that's not true. It's horrible for you. It's actually worse. So, in fact, it's the worst fat you can eat. So trans fat is something we don't want to eat. So you're taking vegetable oil and you're adding a hydrogen to it. What would you call that process, just out of curiosity? I'll give you a second and let you think on it. Adding hydrogen to unsaturated fat to make it solid at room temperature, to make it easier for you to use in baking and such. What would you call it? Well, it turns out we call it, I know it's crazy, hydrogenation. Hydrogenated. If an unsaturated fat has hydrogen added to it, it's been hydrogenated, or hydrogenation is the process. So adding hydrogen to an unsaturated fat to make it saturated, so you can use it in places like baking. You can use it to spread on your butter easily, on your bread easily, and it's not saturated fat, but it is saturated fat. It's just been done chemically. So you take vegetable oil and turn it into solid form. And people thought it was safer, but today we know it's not. All right, that's it.